Start?
Sure.
Okay. So hello everyone. Good morning, afternoon, and evening to everybody. My apology if you guys already start your, the holiday schedule. I know some of you guys already entered the holiday seasons. Sorry to keep you guys busy. Today we are very pleasant to have Dr. Joseph Tamborski to give us the fourth talk this semester. Joe is a coastal hydrogeologist from Department of Ocean and Earth Sciences from the University of Old Dominion University. He got his bachelor degree in 2012 from the University of Buffalo and the major of environmental geosciences. And his PhD at Stony Brook University in geosciences in 2016. Then he did his postdoc at Lagos with Peter Wimbeck from 2016 to 2018. After that, he worked with Matthew Schratt from 2018 to 2020 as a postdoc fellow. Then he got his current position as an assistant professor at Old Dominion University. His interest is how groundwater flow impacts marine and aquatic environments by using geochemical tracers like radium and radium isotopes and the geophysical tracers like H to quantify groundwater surface water interactions in tidal rivers, wetlands, estuaries, and the continental shelf environments. Today, his talk is about the surface water groundwater exchange as a sink of blue carbon in tidal wetland. Okay, Joe, time is yours now.
Thank you, Bocham. Thanks everyone for either waking up early or staying up late. It's great to have this community going forward and keeping these talks up. So hopefully I'll share my screen here. And let's see. Things are not working, of course. Can you see this?
Yes.
Okay. That's good. Seems like it froze. Stop sharing for 1 minute.
Take your time.
Let's try that again. All right. How's that look?
Yeah, looks good.
Great. Oh, thank you again, Coach Out, for the invitation and the introduction. A lot of this work would not be possible without a team of people behind it. So those at Hui, Matt, Paul, and Alec, at USGS, Megan, Eagle, Kevin Kroger, at Dalhousie University, at Barrett and Julia, and then again, a lot of other people who've been really supportive throughout this process, including a fellowship from the Ocean Frontier Institute. So why wetlands? Why tidal marshes? Well, these are really great ecosystems for a variety of services, from habitat and supporting biodiversity to buffering against storm surges. And of course, they take up a lot of carbon dioxide from the atmosphere. So all of this vegetation that you see is regenerated each spring. And so I think traditionally hydrologists or hydrogeologists have thought of these as maybe less interesting because they are very fine grained. But if you actually look down core into these peaty sediments, you'll see that there are lots of buried roots and organic material that helps increase the permeability of these marsh systems. And so to further exemplify that point, I'll play a short little video here. This is a tidal marsh in Massachusetts, US. And what you should be able to see and hear is pour water draining out of this peat. So it's a really muddy, fine-grained system, and I'll play that one more time just to exemplify the point here. The white that you're seeing is actually sulfide oxidizing bacteria. And so these are, in fact, quite permeable systems. There's a high capacity for groundwater surface water exchange. And these marshes are prevalent globally, perhaps most focused along the US East Coast and Gulf coasts, along much of China and Northern Australia by this recent review by Payzin. And these are really concentrated, these marsh ecosystems in mid-latitude regions. And so for all of their ecosystem services, the one that's receiving the most attention as of late is their capacity as a blue carbon sink. So whether we're talking about marshes or mangroves or even seagrass meadows, the idea here is that all of these ecosystems bury organic carbon, and some of that carbon in either the inorganic or organic form, dissolved components, are transferred laterally or outwelled to the adjacent estuary and coastal ocean. And so the thought is that this outwelling or transfer of this dissolved constituent is comparable to, if not exceeding, the magnitude of carbon burial in these blue carbon habitats. And blue carbon is not to say that the carbon is colored blue, right? But just to signify that this is taking place in the coastal system. And so there's been a lot of effort focused on quantifying carbon stocks in sediments in these different blue carbon habitats and quantifying burial rates. We as a community are lagging behind burial estimates in determining this lateral output. This is not entirely a new concept. It dates back to the 1960s, but I'll focus on this schematic of a marsh cross-section here by Wei-Zheng Kai. So the idea here, this marsh CO2 pump, is that these plants take up CO2 from the atmosphere as they achieve photosynthesis. Some of that carbon is blown back to the atmosphere, but some of it is buried. And it's through both aerobic and anaerobic respiration, or the decomposition of organic matter in the peat, that generates DIC and alkalinity, which can be flushed into creeks, flushed into estuaries. And I'll note here this advective transport of pore water, right? This is not an SGD community that's come up with this figure, but that's the idea here, that it's either groundwater or this pore water exchange that is carrying these dissolved constituents into the coastal system. And so just to touch on this idea briefly, this is from my colleague Dave Burdage at Old Dominion. And so if we look at the ecological redox ladder, aerobic respiration, methanogenesis do not produce much or any alkalinity as respiration byproducts, but it is denitrification, manganese, iron reduction, and sulfate, which do produce alkalinity. And so sulfate, there's more or less an infinite pool of sulfate available for microbial respiration in these really anoxic systems. And so that's key to this process, is that the inorganic carbon that is ultimately exported in these systems can remain in the form of bicarbonate and not CO2. So it is potentially viable to stay in the ocean for relatively long timescales, comparable to that of the timescale of carbon burial in these systems. And so a really fantastic synthesis and data analysis study by Ray Najar and Global Biogeochemical Cycles that came out in 2018, looked at the total carbon budget for the East Coast of North America. So looking at the South Atlantic Bight, the Mid-Atlantic Bight, up through the Gulf of Maine, and they broke each system up into 3 boxes: tidal wetlands, estuaries, and shelf waters. And so in each of these components or systems, they can all take up carbon from the atmosphere. They can all bury carbon, and there's all cycling internally. Now, the key takeaway here, and I think it was a surprise to many of the authors, was that about 80% of the carbon that's taken up by the atmosphere in tidal wetlands on the East Coast of North America is actually exported laterally to the adjacent estuarine waters and shelf waters. Only about 20% of that is buried. And this lateral transfer is of the same order of magnitude of the amount of carbon buried along the entire continental shelf. The shelf is orders of magnitude larger than the area that these wetlands represent on the East Coast. So despite their relatively small area, they have a high carbon sequestration potential and a high potential for this dissolved lateral export. And so this is at the regional scale. If we look into the local scale here, coming back to our first image, this is a tidal cycle of pH, DIC, alkalinity for 2 different seasons at Sagelot Pond in Massachusetts. And so what you're seeing here is at low water levels in gray, you have your highest DIC and alkalinity, lower pHs, and at higher water elevations or high tide, you have lower DIC, lower alkalinity, higher pH, and that's repeatable from 1 month to the next. Of course, it varies from spring tide to neap tide to the summer to the fall, but this is a kind of a repeatable concept. And this same group from Woods Hole, instead of doing this now discrete bottle measurements, actually took this a step further, and they developed a sensor called Chanos, which can measure DIC concentration at essentially 10-minute time steps. And so here's a deployment of this instrument over a month-long period. And so you can see really high resolution variability in inorganic carbon concentration. And if you know the flow of water through a creek or a channel with an ADCP, then you can reliably get at carbon import or export fluxes. And so the key takeaway here for me is that this tidal variability mimics a term I think we're all familiar with in this group, and that is tidal pumping. And so there is, to me, the need to think of this not just from a surface water perspective, but from a subsurface perspective as well. And so that's kind of where I'll be going with this over the next 20 to 30 minutes, is looking at this from more of a hydrogeologic perspective. So these systems, those fine grain organic rich sediments, that's called peat generally, and that peat usually sits on top of a sandier unit, a coastal aquifer, if you will. So in terms of thinking about SGD, right, our groundwater is flowing through our coastal aquifer. Some of that may flow through the peat, discharging into tidal channels. If a channel is cutting through the peat, that can discharge directly to the channel. But then you have the flooding of the marsh or the flooding of the peat at high tide that helps recharge the peat. And there's some drainage at low tide or sometime thereafter. Now, if you have a storm event or a king tide, you flood a greater percentage of your marsh creek shed. And so it's not perhaps quite as nice of a cycle as a cartoon depicts, but you've got these, variable length and spatial scale flow paths in these systems. And the kind of the classic terminology here would be low marsh, representing lower elevations, more frequently flooded, and high marsh. So that corresponds both to vegetation zonations and flooding frequency. So the key questions that we're really asking here is how does surface water, groundwater exchange modulate this carbon flux or this carbon outwelling in the coastal zone? What is the importance of submarine groundwater discharge versus the drainage of these marsh peats, which I'll refer to as pore water exchange or pore water drainage. And then our last question then will be, exactly where is this marsh peat being flushed? And so the framework that I'll be presenting is for this site, Sage Lock Pond, where we have a relatively thick peat near our channel of about 1.5 meters to 2 meters in thickness, and it thins as you go towards the upland transition zone, where there's more freshwater tolerant vegetation. In the low marsh, which is where we're focused, it's Spartina alterniflora, and we'll be looking at a transect from basically near the creek towards the interior of this low marsh system. And so this is a micro-tidal system. It's about a 0.5 meter tidal range on the south shore of Cape Cod in Massachusetts. And we looked at 2 different creek sheds, this one in red and this one in blue. I'll primarily focus on this one in red here, where you've got the upland, higher elevation from this LiDAR image, and then low elevation in the near the creek, near the estuary. And so here's that transect going from the near creek towards the interior of the low marsh here. And this is a great site to work because if you've done any work in wetlands, it can be quite hard to walk around, but there's a really nice catwalk out here that allows you to access the marsh when things are flooded. And this is a rare case. This system is not usually flooded to this extent, but both strong winds that blow from a particular direction coupled with rain can flood this marsh from time to time. What it often looks more like is something like this, where you could walk around if you wanted to. And so we collected surface waters or creek waters where we had a YSI in ADCP deployed for several months at a time. And we collected pore waters with a push point piezometer. So we did a lot of radium isotope work, DIC, alkalinity, sulfide, anions, cations, you name it. We tried to measure it. And we also collected shallower pore waters with sippers. So kind of a syringe-based approach to get at really small scale variation with depth. I will note here, I tried to measure radon on a few occasions. There's virtually no radon in this pore water of the peat. There's really low radium-226 content, and it just is not present as an end member. So here we focus on radium. And so we collected radium both in the pore waters and in the tidal creek here for 16 or 17 tidal cycles over multiple years. And here's a summary of some of that radium tidal creek data. This is showing you 224 in green, 226 in red, and 228 in blue. And they're partitioned here by whether they were collected on an ebb or a flooding tide. So ebb tides as hollow symbols. And what I want to emphasize here is that there's a lot of variability as a function of water elevation within the creek channel, and also as a function of salinity. But there's a statistically significant relationship with increasing radium activity with decreasing water elevation, kind of matching this idea of tidal pumping in the system as a conveyor of groundwater and pore water. And for the long-lived isotopes, 226 in red and 228 in blue, you do see increases in these isotopes with decreasing salinity, just pointing towards a brackish end member. You do not see that for radium-224, which is a factor of 5 or so greater than radium-228 in the same decay series. And its maximums are observed at creek water salinities, not at brackish salinities. And so it's a lot of data here, but what's really great about this system is that it fits rather nicely into a 3 end member mixing model. So here's the same creek water samples, now bound by our coastal ocean end member Vineyard Sound, our brackish SGD end member, and then our marsh pore water. And this is looking at the ratio of the short-lived radium-224 to the longer-lived radium-226. And this is possible because seawater is continuously recirculating through the peat, and so there is a lot of radium-228 and thus thorium-228 that's in the peat that allows this enrichment of radium-224 that is not present in the sandy aquifer below. And so even better for us is that this relationship more or less holds up if we look at this versus our DIC content of our end members. And so this is really what we're trying to track, which helps give an extra degree of confidence in some of this. And so we can take this end member mixing approach, and because we have a ADCP in our tidal creek, you can get at the fraction of each member and simply multiply it by your water flux to get at a volumetric contribution of marsh pore water versus brackish SGD. So looking at any one tidal cycle here, showing you water elevation as the gray line versus the radium isotopes. And you'll see that there are characteristic peaks at ebb or near low tide and lower activities at higher water levels at high tide. Key to this, though, is that they do not necessarily all peak at the same time. Radium-224 here, for example, peaks later than that of radium-226. Now, taking this end member mixing model approach, what those fractions would look like over the same tidal cycle is that, you're anywhere from 90 to 100% seawater at high tide, but at ebb tide here, we've got about equal proportions of marsh pore water mixing with seawater in this creek. So again, if you have your ADCP in the creek, half that water should be derived from marsh pore water, half of it should be just this tidal exchange of seawater. And so this idea of both greater pore water drainage at ebb and low tide is not surprising. Alicia Wilson's done a lot of great modeling work to show this, but we are happy to confirm it and see it at our study site. So with that said, I don't want to belabor it, but we did 17 tidal cycles. A lot of this was led early on in 2015 by Megan Eagle at USGS, and I continued it on in 2018, 2019. And so I only want to emphasize really one thing here is that we collected a ridiculous amount of radium data, but we have seasonality here. So these are spring seasons in green, summer in red, and fall in blue. And so each one of these more or less represents a spring tide and a neap tide tidal cycle sampling event for each period. So we can look at kind of minimum and maximum of water elevation over the course of 3 years. So of course we want our end members if we're going to look at carbon flux. And so this is now pore water profiles, depth below the marsh surface corrected for elevation. So as we go from near the creek towards the interior of the marsh. And what we see is that the pore waters at this site, they're all very saline, with the exception of the profile collected closest to the creek, which actually shows this brackish SGD influence at depth. And I'll note here that this orange line is the peat sand contact or interface. So this is in the sandy unit below. And the pH is low, but not surprising given the type of system we're in. And the DIC content is really quite high. But a key takeaway is that the profile near the creek is about a factor of 2 lower than that of the interior of the marsh, which is suggesting that it's being flushed more frequently or at shorter pore water residence times near the creek. And these profiles are fairly reproducible from one season to the next. So if we look at the fall season, just a few months later, we see the same salinity profile, similar ranges in pore water pH and DIC content. And so with this, we have a pretty good idea of what the carbon end member should be as we look at determining flux from SGD, or marsh pore water drainage. And so taking those 17 tidal cycles that we had and kind of summarizing it into one figure here. This is just looking at water exchange for the spring, summer, and fall seasons for those 3 different time periods. And so interestingly, it does appear that 2019, we were estimating higher water exchange rates. The one driver we could come up with was that this seems to track variation in mean sea level at the site. And so the idea here, which requires further testing and validation, this is, while it's a lot of radium data, it's a limited number of seasonal and annual points, but the idea is that if you flood a greater proportion of your marsh during higher periods of mean sea level, you're recharging more of the marsh, which corresponds to more discharge. You're also adding more sulfate to the system to allow for more anaerobic respiration and generation of this DIC. So all of that's to say, I would take it with a grain of salt, but it is statistically significant. But our pore water exchange, anywhere from 280 to 1,280 grams of carbon per square meter per year. The brackish SGD is about an order of magnitude less. It's 30 to 80 grams of carbon per square meter per year. So the drainage of this marsh peat, really the dominant conveyor of inorganic carbon at this particular site. There's evidence here that it's, of course, seasonally variable, but temporally variable on annual timescales. And so I think there's probably a tipping point associated with that. If sea level continues to rise or you have variability in mean sea level, there will be a point where you just flood the marsh so much that there's a lack of exchange. But this is something we're interested in continuing to study. So that brings me to the kind of next question here, which is, well, where's the carbon derived from in the marsh peat, or where's the peat actually being flushed? And so for that, we're going to turn to sediment cores, where we looked at the disequilibrium between radium-224 and thorium-228. So I got to give a shout out to Ping and Shengming, who answered a lot of questions as I spun up this method. But I think it's a really powerful method, and I would encourage many others to try and give it a go at their own study sites. The idea here is pretty simple. Thorium-228, the 2-year half-life, it's a high KD, it's very particle reactive. And when it decays to radium-224, that radium in brackish and marine systems should be more soluble. So in the presence of advection and flushing, you will lose some of that radium, and so you'll have less radium on the surface exchangeable portion of these particles compared to the amount of thorium in red here that would be present. So this indicates a relatively recent flushing of this upper 3 centimeters of marsh sediment. At depth, where radium and thorium are equal, or they're in secular equilibrium, there would be minimal flushing over the approximately 10-day integration window of this isotopic pair. So that's the idea in a nutshell. And so we looked at cores across a variety of seasons. And so here's just, I'll say what is an interfacial radium flux, meaning what's happening in the upper few centimeters of the peat for winter, spring, summer, and fall seasons. And what we do see is when the marsh is flooded more than 20% of the time, there seems to be an increase in this interfacial radium flux. So there's more export, more cycling of pore water in the upper few centimeters when the marsh is more frequently flooded. This kind of ties back into this idea of the water exchange being correlated with the variability in mean sea level at this particular site. But this is just the upper few centimeters, and the peat here is about 1.5 meters to 2 meters thick. So we really want to get the whole story. So had a great summer student, William, help me out here. And so we collected some pretty deep cores using a Russian peat borer here. So we were able to take 50 centimeter chunks at a time, down to this peat sand interface, and would have happily gone past that if we could, but just couldn't get any deeper at that point. But so we collected these cores now, again, right near the creek, and then going through the same transect towards the interior of the marsh. And so here are the radium and thorium activity profiles. Again, this is elevation corrected for NAVD from the near creek, 7.5 meters away towards the interior of the low marsh here. And I don't want to spend a lot of time on the activity profiles here, except to note that this kind of decrease in the upper 20-30 centimeters is actually representing sedimentation and decay of thorium. And there's some mid-depth maxima and bottom depth maxima that may be actually related to groundwater flow through the peat, carrying radium-228 that decays to thorium, which is then locally scavenged. But what is more useful here is to actually look at the activity ratios. And so if you look at the ratio between radium-224 and thorium-228, anything in this pink band is in secular equilibrium. And I realize that this is larger than a thin dashed line at 1, and that's because it's accounting for our analytical uncertainties on both the thorium and radium measurement. So anything to the left of this is in deficit, indicating some recent flushing. Anything to the right would be in excess. And so what we do see now, if we're looking at the near creek site, is this deficit in the upper few centimeters. We have another minor deficit between -20 and -40, and a larger deficit here at depth. So looking at this across the marsh platform, this shallow deficit is observed across the marsh platform, but it diminishes in magnitude as you go towards the interior of the marsh. But this kind of -20 to -40 range is observed to some extent across this particular transect. This deficit at the creek is not necessarily observed in the marsh interior. And so to sum up what I think is going on here, and to put this in the context of carbon, our upper few centimeters are being flushed by some combination of waves and tidal overtopping. We have a zone of equilibrium. We have a zone of spring and neap tidal pumping. This is actually the same elevation horizon where water level in the creek is at its minimum during the course of a given tidal cycle. And so at that depth horizon in the channel, there are actually large macropores that you can see from irrigating organisms. And then at depth, there's a zone of equilibrium. So residence times likely on the order greater than 10 days down to the peat sand contact. Near the tidal creek, things are a little different. Again, we have this disequilibrium in the shallow root zone, this tidal pumping deficit, and then this larger deficit at depth, which corresponds to this salinity transition zone. So there is perhaps this influence of this brackish SGD that's flowing through the peat at this site. So what's nice here is that we can integrate the deficit at different depth horizons, and we know the radium content of the pore water and the DIC content of the pore water at those distinct depth horizons. So we can quantify both water exchange rates and DIC flux, in this case, interfacial. So what's happening in the upper 5 centimeters of the peat, and then at depth, which is just below the active root zone. So the interpretation here, which may require more validation, is that what's happening in the upper few centimeters is a vertical exchange pathway, and what's happening below the root zone, below 30 centimeters, is a lateral horizontal transport pathway. And so looking at these cores over 3 different seasons, we see unsurprisingly higher flux during summer than fall than winter. But what is kind of the key takeaway here is that the upper few centimeters of the marsh peat only contribute 10 to 20% at most of the DIC that is exported from this site. And it's about 50% of the carbon that's exported from just the first 7 meters or so of the of the Near Creek zone. In other words, this more recent recirculation zone driven by more frequent flooding and drainage. And so I did mention we studied 2 sites, and so I want to just quickly introduce the other. This is the same data as DIC flux and water exchange on the top, but now broken up by site 1 versus site 2, where site 1 is in red and that second site is in blue, which was not sampled during the fall. So that's a caveat here. But what is interesting is that during the winter, water exchange rates were comparable between the 2 marsh sites, but the DIC flux for the second site was really through the roof. It was enormous. And that's because there was a factor of 3 higher DIC pore water content at this marsh site. And so that gets us into this question of, well, what's driving end member carbon variability between sites? And so to briefly touch on that, if you look at the DIC and DOC for that matter, content of the pore waters in the marsh as circles or the subterranean estuary in triangles versus this is delta sulfate. Billy published a nice paper in the last year on this topic. So the assumption is that the peat is recharged with a chloride sulfate ratio of oceanic waters, and you can predict the amount of sulfate that should be present from your measured chloride content. And so a negative value here would mean that you have a deficit of sulfate with respect to what's predicted from that stoichiometric ratio. And this is a rather nice 2-to-1 increase of DIC content with delta sulfate, which is suggesting that this is sulfate reduction as the primary anaerobic respiration pathway for generating DIC. And it's these almost 20 millimolar DIC concentrations at our second site that's driving such large fluxes in comparison to our first site, despite similar rates of water exchange. So that was exciting. There's still more work to be done to disentangle exactly what's going on here. But some of this can be true for DOC as well. And so to take those sediment cores and actually flip the script and say, well, not water exchange, but how about residence time? So if we know the depth horizon and we know the porosity of the sediments, we can convert that to a time scale. And I'm just showing here the summer season split out by deep samples versus surface samples. And then there's quite large error bars on here, which is not surprising as you get out to longer residence times. That's going to be a nature of the calculation based on the production rate of the sediments. But what is interesting is that at very low residence times, 1 day or so, the delta DIC, or the difference between the pore water and the surface water DIC, is quite low. And after you exceed 2 days, 3 days, you have a massive increase in DIC, which would suggest that that's the time scale for sulfate reduction needed to generate that carbon. So to put this all together, we have a term called net ecosystem carbon budget, or NECB. At this site, we know that peak annual standing stock is on the order of 2,500 grams of carbon per square meter. Some work by Joe Carey estimated GEP on the order of 700 grams of carbon per square meter per year. So that's counting after the fact for the amount of CO2 that's blown back off to the atmosphere. And this is an estimate for 2014. Carbon burial rates from Megan on the order of 100 grams of carbon per square meter per year based on lead-210 and cesium profile dating. And then ecosystem respiration within the peat on the order of 150 to 200. So our brackish SGD is again around 30 to 80. Our marsh pore water drainage is in 2014 was estimated at 400 grams of carbon per square meter per year. That would make the system balanced. Our radium tidal time series was a range of 300 to 1300 increasing towards 2019. So I note that estimates of GEP or ER, they're likely annually variable as well. But this is encouraging that there's, at least in 2014, some balance in the system. And the key point is that this, even the low end here is a factor of 2 or 3 higher than the amount of carbon buried in this system. So what? Well, the so what is actually a pretty big deal. This is a natural climate solution to mitigating greenhouse gas emissions. And so there's a big push. Kevin Kroger, colleague, doing a lot of this work here. We're fighting to get this lateral carbon flux included in terms of crediting within this blue carbon framework. So if you're a polluter, if you are let's say a transportation company and you emit a lot of CO2 and methane, you may be looking to buy wetlands to offset some of the carbon emissions. Well, that might not be cost beneficial as opposed to some other metrics or methods that are available to you. But if you increase the value of your wetland by 2 or 3-fold by now including this lateral flux, all of a sudden, both the benefits of CO2 drawdown are maximized, the financial costs are made more palatable to the buyer, and that doesn't even begin to document the other benefits of habitat restoration or storm surge protection that these particular wetlands provide. So there's a push to make this lateral carbon flux a part of this carbon crediting framework. And I love this slide from Matt Bogard in Global Biogeochemical Cycles, which looks at lateral carbon flux, which is very limited for tidal wetlands, just 4 studies here, versus the amount of net ecosystem production for the system. And these lines indicate how much of that production is actually lost laterally to the adjacent aquatic or marine ecosystem. And so the takeaway here is that coastal wetlands are among the least efficient ecosystems for the storage of fixed carbon. So they're taking up a lot of CO2, but they're also exporting a lot, and it's really understudied. And so Julia and I put together a little data synthesis review article. And so this is a summary of studies along both the East Coast of the US as well as globally. Any study that we could find a area normalized estimate. And so the East Coast of the US is decently well documented, although only 2 or 3 of these sites actually have lateral carbon export estimates. So this is including both brackish SGD and then pore water exchange as the color scale here. And so there is a lack of any studies at larger tidal ranges, and again, very few studies that actually quantify carbon export. So I'm going to make a call to arms plea here and say that we have a lot of studies in our community looking at nutrient cycling and nutrient export, and we need more studies on these tidal systems and on carbon transport. So I'll put my money where my mouth is and just briefly introduce some of our new local work, zooming in to the Chesapeake Bay region here. So we're looking at southeastern Virginia. This is all work that's taken place over the last few months now. It's been a while to get the new laboratory spun up, but this is a really excellent, I think, ideal laboratory for starting to ask some of these questions. This is First Landing State Park. It's the first landing of the English settlers back in the early 1600s before they decided that this place was not good for them and they went up the James River. But here there's a nice water table dome, so a max head of about 2.5 meters above mean sea level. And that's just simply set by the rate of recharge of the system, the hydraulic conductivity, and the thickness of the island. So groundwater flows out radially of this dome towards the Chesapeake Bay, towards the Atlantic Ocean, and towards Broad Bay here. So down in the southeast here, Broad Bay, is where we have this really magnificent ghost forest. And so for those of you who may be unfamiliar with the term, ghost forests are essentially ecosystems in transition from forested uplands to emerging marshes. They're being inundated by salt, and so there's a transition from salt intolerant to salt tolerant species. And so the name ghost coming from the dead trees that are littered across the ecosystem. So we just in September installed a set of wells kind of at the upland, and a nested set, and then a lower down gradient set of wells as well. And so I'll take a break from radium isotopes for a minute and talk more about traditional hydrogeologic methods. We had an undergraduate student who installed some of these wells just by hand augering. And again, you can see the dead trees kind of scattered throughout the ghost forest. So a spooky place to be working. This is more typical of the conditions. It's not always flooded like that drone image I'm showing you. And so here's the variability in head, and these were pulled just a few weeks ago, so the student could write their senior thesis, but we're hoping to get these loggers put back out in the new year. So our upland well is responding moderately to precipitation, and then our wells in the ghost forest are actually much more driven by not precipitation in gray, but by the variation in the tides in this kind of teal color. So maximum heads syncing up pretty nicely with our maximum tidal water levels. And so looking at two of these wells, our up gradient well and our down gradient well, we see their salinities, our down gradient wells, similar to creek water salinities for much of this month-long deployment. And our upland well, which has a salinity of about 3 PSU, does have these notable increases discretely over the course of this month-long monitoring period. And so looking at the gradient, the hydraulic gradient between these two wells, anything above this red line, the gradient is going towards the creek or towards the sea. Anything below it is going towards the land, towards the upland. And so it's actually these periods of essentially spring tides when the gradient is strongest and it does reverse towards the land. And this is here, again, over here, corresponding to these brief periods in time where there is salt being intruded to the upland of this ghost forest system. So not observed during neap tide, smaller tidal amplitude periods. We have a lot of work to do here. We have not done any carbon or alkalinity or radium or radon work yet, but we want to get the plumbing right before we continue on with our carbon flux accounting. And so I'll end on this more thought-provoking slide than anything else that Julia and I put together, which is to kind of make the plea or the case for including hydrogeology within the context of these coastal systems. If you are studying vegetation or studying climate variability or studying geomorphology, is that one, we should be encouraging interdisciplinary work. One of these is, certainly impacting the others, and that we want to really create a holistic framework for studying these wetland systems. That hydro is so important in driving vegetation zonation, but this is of course so important in stabilizing the morphology of the system, which feeds back into the hydrogeology of the system. So for those who are continuing this work, I can certainly encourage taking this team-driven holistic approach. And I'll just conclude by saying, if you're to take anything away from this, it's that we need more studies looking at carbon flux in these tidal systems. And then our two big summaries for our Sage Law Pond site, that the bulk of the carbon is exported from this sediment horizon intersected by mean low tide. And we saw an increase of exchange as mean sea level was increasing. But, you know, to what maybe tipping point, I'll say, is that the case? And so thank you, Bo Chow, for the invitation. Thank you all for your attention, and I'd be happy to take any questions.
Thanks, Gio. Very nice work, very nice talk. Okay, now it's time for questions, comments. If you do, you can just, yeah, Lisa.
I have a sort of unformed question, which is, the first thing I want to say was thank you for the really nice talk. And you have some really great field sites where you can actually see through salinity, the groundwater salinity is different from the salinity that's circulating in the marsh sediments near the platform. It's so nice to have a real tracer that says which one is which. All my sites are just like totally saline by the time you get out to the salt marsh. So that's leading to my unformed question, which is, for me with salinity, I've seen DOC is just way, way higher, and I've seen this in other people's papers in this room here, where the DOC of fresh groundwater and below the uplands is just way higher than the DOC in the saline salt marsh system. And so that got me interested in when you're having, you know, storm events, how this actually gets out. But the overall question, because I am not a carbon person and I have a lot to learn, is between DOC and DIC, do we need to know about DOC? Everyone focuses so much on DIC.
Yeah, thanks, Alicia. It's a really good question. I did not focus on DOC much because DIC is the dominant carbon conveyor. But we have a lot of DOC data from our Sagelot Pond site. Kevin Kroger and Megan Eagle have done a lot of work on that. The flux is about an order of magnitude less than the DIC flux, but the brackish groundwater, or the terrestrial groundwater, in that case is a much more important driver than the pore water drainage is. And it's exactly that. It's the fresh water is more enriched in DOC through the leaching and transport along the land ocean continuum, and it's not necessarily the same source in the peat itself.
Cool, thanks.
All right, Henry.
Yeah, thanks, Joe, for your great talk. I do have a question with the really cool data you were showing on sort of like the, gosh, this shows my ignorance, but the ratio you were using to show like kind of like almost like a turnover rate or renewal rate of the pore water in the peat sediments. That sort of deeper layer where you said it seemed to be tidally influenced, do you have any information, and I'm sorry if I missed this, on like how the depth of that pretty thin layer, because I think it was only like a couple decimeters, how it corresponds with the height of like water table fluctuations? And maybe this is what you're saying, but I just wanted to understand it because I thought it was cool and might have cool implications for what we're thinking about with water table fluctuations and carbon cycling.
Yeah, so unfortunately, we put in 2 wells and one of the data loggers didn't record any data. So we don't have a complete picture on the water table variability at that site. But the idea in that case was that 20 to 40 centimeter elevation range was essentially the depth range where the creek levels at its lowest during low tide. And if you were to walk that catwalk, you would see all of these macro pores in the peat there, which is like, likely where it's being flushed and exchanged. But yeah, certainly going forward, trying to tie this in better with the hydrologic monitoring is a goal.
Okay. We have a question from the gentleman, Song Jiehe, asked the question, 2 questions. The first is, how did you locate the peat to sand interface by getting deep cores?
Yeah, so
Second is yeah, go ahead.
that one's easy. It's just by feel with the push point piezometer. If you push it in, it's going in like butter, and then you feel a lot of resistance, and so just marking off that depth.
Okay, the second is, if you have a radon in the member in your pore water, will you still use the radium 226? I think it's a radium 224, thorium 228 method. The later seems to require lots of M power and the redac.
I'm not sure I fully understand the question. I will say again that there was very little radon in the marsh pore water. There's not a lot of radium 226 of this peat. And you are right, the radic for those sediment cores does require a lot of effort. Those core sections all had to be measured within 12 hours of collection, and fortunately Matt and Megan at Hui have 16 counters. So but I realize that not everybody has that capability. But if you're here and you can rephrase your question, I could answer it more clearly.
Hi, I don't know if you can hear me. So I was asking, basically you answered my question kind of. So I work in Louisiana, in the salt marsh. So the salinity there is a lot lower than sage pond. So I think depending on season, sometimes even in pore water, the salinity gets pretty close to 10 or lower. Even it's very typical salt marsh vegetation like spartina and jungus. So and if I understand it correct, the radium 224 to thorium 228 method require the salinity to be higher. So and then also, like you said, you had lots of resources for your research. And in my lab, we only have 4 radic systems. So we didn't think that we could use your method. So for me, I tried to use radon. So the radon in my pore water is also reasonably low, especially during like summertime when the marsh gets flooded daily. So I had like right now I have collected data at 5 different seasons, but I'm having a lot of hard time interpreting it. So I'm wondering if that could be due to the fact the radon and member is not quite there during certain season. But anyway, thank you though. It's a very useful talk. I'm glad I got up at 6:30 to try to listen.
Yeah, thanks for having your coffee with us this morning. Yeah, certainly the radium method would not work as well in a freshwater or low salinity marsh. But having maybe some other tracers or complementary methods is always worth pursuing.
Joe, actually, I have a follow-up question. So when Pinghe got the method, the disequilibrium between radium 224, thorium 228, he used it to calculate the PX fluxes, right? The pore water exchange fluxes. And I noticed that you also calculated the pore water exchange fluxes. I assume you're using the mass balance model by using different tracers, right?
Yeah,
Are they comparable?
So Ping and Xingming published a paper 2018, 2019 in LNO. It was a 2-layer transport model, and that's essentially what we're using here. So the idea that the upper peat is being flushed vertically, and then below some depth horizon, things should be transported laterally. And so we're standing on their shoulders here, so to speak, for using that method at our site.
No, I meant the fluxes, the px of fluxes you got is based on the mass balance model, right?
Yes, so in integrating that radium thorium deficit, you get a flux, and then yes, having the pore water end member for that deep horizon, you can get a mass balance approach to quantifying exchange.
I noticed that you used a 3 end member,
Mhm.
3 end member model, and you got the SGD fluxes and the px fluxes.
Right.
That is not based on the radium and thorium disequilibrium method. That is not, right?
No, that one is based on the Tidal Creek radium isotope measurements.
Yeah.
So
Yeah,
we
and
had
you can also get the px fluxes based on the disequilibrium method.
right. Okay, I see what you're saying. So yeah, they agree reasonably well, the two different approaches.
Okay.
I didn't touch on it here, but we also included Alex's multiple linear regression model approach for quantifying DIC flux in an effort to figure out what method is tracing what. And they are all looking at different spatial temporal scales. The creek-based approach for DIC, you might lose some CO2 by degassing. The radium thorium approach, you're integrating over the mean life of radium thorium, and the Tidal Creek radium approach is more instantaneous, right? And so it's not exactly apples to apples. So there's disagreement at some point, but having multiple methods, I think we start to converge on a similar answer.
Okay, Qian, your turn.
Okay, hello. So thank you for your talking. Yeah, so I have a preference. Yeah, so how to explain the phenomenon that the radium-24 is high than the thorium-28? Yeah, because I saw this phenomenon in your fieldwork. Yeah, so when you estimate the fluxes, including this part. Yeah, okay.
Yeah, so great question. There are some depth sections where there was an excess of radium compared to thorium.
Yeah.
And the most likely explanation there, and it's mostly in the shallower sediment sections, is that those are probably zones of active manganese cycling. So if you have some active manganese production or reduction, that can adsorb any radium-224 that's being advected away, creating a local excess, essentially.
The radium? Okay, thank you.
All right, any more questions? Yeah, so there's a lot of actually there's a lot of reports. The SGD or the PX literally transport the DIC or alkalinity into the ocean, and could this fluxes be observed in the ocean side?
So I think that's a big question mark and unknown in our field is, where does it end up? Is it staying in the estuary? Is it making it out into the open ocean? So if somebody wants to write a proposal and we can work on that. But that's not a trivial thing to get at, right? So there's a lot of CO2 loss, there's internal cycling, there's some burial. It remains to be seen exactly where the ultimate fate of that carbon is. And I think that's probably the big thing that gets us over the ledge to get this included in terms of crediting and incorporation into policy. So if you can prove or have a better idea as to where it's actually going, you're more likely to meet that question when you talk with policymakers.
Yeah. Okay. I think that's all for the questions from the audience. Thanks again, Joe. Thanks. Very nice
Thank you
talk.
all. Yeah. Happy holidays.
Yeah, happy holidays. Oh, Stephanie have a question.
Actually, I was just clapping, but it was just reacting
Okay.
to clapping. But thanks, Joe. That was a great talk.
Thanks, Steph.
guys.
All right.
All right. Bye-bye, you guys.
Take care.